Hello students, welcome to E-PG Pathshala program. I am Dr. Shoaib Lukman from the Council of Scientific and Industrial Research, Central Institute of Medicinal and Aromatic Plants, Lucknow. Today we are going to talk on the biosynthesis of lipids part 3, which is an integral part of the paper Lipid Metabolism. The objectives of the paper include the understanding of the biosynthesis of steroids. Steroids are important molecules as far as derived or precursor lipids are concerned. What are the implications of the cholesterol biosynthesis, and how do cortisol and testosterone synthesize? Coming to the biosynthesis of steroids in animals and other living organisms, this conduit is being carried out in discrete ways, making it a frequent target for anti-infective agents and other antibiotics. For example, the statins, the cholesterol lowering drugs, targets the steroid metabolism. In this metabolism, the acetyl-CoA in the mevalonate pathway act as building blocks that configure isopentenyl pyrophosphate and DMAPP, followed by the formation of lanosterol, and this lanosterol is the first steroid to be synthesized. In this module of the biosynthesis of lipid, we study the synthesis of branch-chain fatty acids, how these branch-chain fatty acids are synthesized from the acetyl-CoA, and how the branch-chain alpha keto acid decarboxylase enzymes work to form the branch-chain fatty acid. In addition to this, the fatty acid synthase system operation also be looked into. Just continuing with the biosynthesis of lipid, part 2 explain these conundrums. The additional modifications in the steroid biosynthesis, especially the lanosterol, gives rise to the ensuing steroidogenesis. Steroidogenesis is the natural course of action by which steroids are enkindled from cholesterol and gets altered into several other steroids. the biosynthetic pathways of steroidogenesis vary amid distinct species, but in humans, the products of the biosynthetic pathways of steroidogenesis include aldosterone, androgens, corticosteroids, cortisol, estrogens, progesterone, and testosterone. Let's discuss the biosynthesis of cholesterol. Cholesterol is predominantly an indispensable molecule of the class sterols, containing a hydroxyl group and 4 fused rings. Being an important constituent of lipid membranes, cholesterol also act as a predecessor to numerous steroid hormones comprising cortisol, estrogen, and testosterone. In animals, cholesterol produced with a relative speed anecdotal to the type of cell and function of the organ. In liver, approximately 20 to 25% cholesterol production occurs every day. Adrenal glands, intestine, and reproductive organs are the other sides of higher synthesis of cholesterol. Cholesterol is also synthesized from acetyl-CoA through numerous conversions. These steps in cholesterol biosynthesis include the formation of acetoacetyl-CoA from the acetyl-CoA. It takes place when acetyl-CoA converted to acetyl-acetyl-CoA and further dehydrated to hydroxymethyl-gluteryl-CoA. The reaction was catalyzed by thiolase, which converts acetyl-CoA to acetoacetyl-CoA, and HMG-CoA synthase, which results in the formation of HMG-CoA. The HMG-CoA is reduced to mevalonate by an enzyme called HMG-CoA reductase. Remember, this is a rate limiting step as well as the regulatory step. which is irreversible in cholesterol biosynthesis. The competitive inhibitor of HMG-CoA reductase, which is statins, target this step for their action. And further, this mevalonate formation from HMG-CoA is further converted to HMG MVA5 phosphate and MVA5 pyrophosphate with the help of an enzyme called mevalonate kinase and phosphomevalonate kinase respectively. The intermediary mevalonate 5 pyrophosphate decarboxylase enzyme converts this mevalonate 5 pyrophosphate to isopentenyl pyrophosphate. And this step of the multi-sequence reaction requires adenosine triphosphate as an energy source. The formation of isopentenyl pyrophosphate, 3 moieties, then further condenses to structure the formation of geranyl pyrophosphate and farnesyl pyrophosphate. The conversion of isopentenyl pyrophosphate to geranyl pyrophosphate takes place by geranyl transferase, and isopentenyl pyrophosphate conversion to farnesyl pyrophosphate takes place in presence of enzyme farnesyl pyrophosphate synthase respectively. The formation of IPP isomerase or the presence of IPP isomerase converts the formation of isopentenyl pyrophosphate to DMAPP, which also results in the further formation of geranyl pyrophosphate and farnesyl pyrophosphate. Remember, this geranyl pyrophosphate and farnesyl pyrophosphate step is the key target step for the bisphosphonates compounds. These bisphosphonate compounds inhibits the formation of GPP and FPP. Furthermore, FPP2 moieties subsequently deliquesce to create squalene through the deed of squalene synthase in endoplasmic reticulum. So the formation of squalene through squalene synthase takes place with the assemblage of 2 molecules of FPP. Also, the FPP forms GGPPP, geranyl pyrophosphate, by the action of geranyl pyrophosphate synthase enzyme, which further give rise to prenylated proteins. In the final steps of the cholesterol biosynthesis, squalene monooxygenase converts squalene to 2,3-oxidosqualene and squalene epoxidase or oxidosqualene cyclase further cyclizes squalene to structure the formation of lanosterol. Ultimately, lanosterol is subsequently transformed to cholesterol. The entire pathway of the cholesterol biosynthesis is shown on the right side of the slide, which explains a multi-sequence complex reactions going on in the organisms that synthesize cholesterol. In 1964, Konrad Bloch and Fyodor Leinen shared the Nobel Prize in Physiology and Medicine for their discoveries pertaining to the regulation of fatty acid metabolism and cholesterol mechanism. In general, the cholesterol biosynthesis occurs in three stages. The stage 1 occurs in the cytoplasm, whereas stage 2 and 3 takes place in the endoplasmic reticulum. The stages of the cholesterol biosynthesis are: first, the biosynthesis of isopentenyl pyrophosphate, which is the building block of IPP or isopentenyl pyrophosphate. The second step is the genesis of squalene through the abridgment of isopentenyl pyrophosphate. The isopentenyl pyrophosphate serves as the building block of the cholesterol. And the third stage includes the translation of squalene into cholesterol by a number of enzymatic reactions. The regulation of cholesterol biosynthesis. It is unswervingly regulated by the level and the concentration of the cholesterol present, albeit the mechanism of homeostasis involved are merely understood. A high intake of cholesterol-rich foods leads to a decline in endogenous production, whereas lower intake has the pugning outcome. Sterol regulatory element binding protein 1 and 2, which is abbreviatedly known as SREP-P, is the main regulatory protein that sends the intracellular cholesterol level in the endoplasmic reticulum. In the charisma of cholesterol, two other proteins, namely SREP-P, cleavage activating protein called SCAP, and INSIG1, were found bound to SREP-P. When the level and the concentration of cholesterol goes down, INSIG1 separate from the SREP-SCAP complex allowing it to drift towards the Golgi complex, where site 1 and site 2 proteases cleaved SREP-B. The site 1 and site 2 proteases get activated by SCAP when the concentration and level of cholesterol becomes low. The emitted SREP-B further transfers to the nucleus and work as a transcription factor to truss with sterol regulatory element, which kindle the transcription of numerous genes like HMG-CoA reductase as well as low-density lipoprotein receptors. The HMG-CoA reductase leads to an enhancement of endogenous cholesterol production, whereas LDL receptor scavenges flowing LDL from the bloodstream. In 1970s, Dr. Michael Brown and Dr. Joseph Goldstein clarified this conundrum, and they received the Nobel Prize in Physiology and Medicine for their effort in 1985. Subsequently, they showed the expression and regulation of numerous genes by the SREP-B pathway that manages lipid formation and metabolism and its allocation in body fuel. High level as well as concentration of cholesterol shuts off the biosynthesis. The enzyme HMG-CoA reductase includes a membrane and a cytosolic domain liable for its catalytic activity. The membrane domain functions to signal for its degradation. Increase in the level and concentration of the cholesterol or other sterols effectuate a change in the oligomerization state of the domain that makes it further susceptible to proteasome annihilation. The enzyme activity can also be diminished by adenosine monophosphate activated protein kinase phosphorylation. When ATP hydrolyzed, adenosine monophosphate is formed, which activate kinase functions, and cholesterol biosynthesis remains halted as long as ATP level becomes low. Cortisol biosynthesis. In humans, cortisol, a glucocorticoid class of steroid hormone, is produced by the zona fasciculata of the cortex region of the adrenal glands The cortisol is also synthesized from cholesterol. The cortical region of adrenal gland, especially zona glomerulosa, produces aldosterone. Zona reticularis produces sex hormones, while medullary region of the adrenal gland lies beneath the cortex, primarily secretes the catecholamines, namely epinephrine or adrenaline and norepinephrine or noradrenaline under the considerate stimulus. The cortisol biosynthesis is invigorated by the adrenocorticotropic hormone ACTH secreted by the anterior lobe of the pituitary gland. The production of ACTH is visualized and vitalized by corticotropin releasing hormone, which is discharged by the hypothalamus. Increase in adrenocorticotropic hormone concentration increases the cholesterol level in the inner mitochondrial membrane through the regulation of the steroidogenic acute regulatory protein, which is also called as star protein, thereby stimulating the major rate limiting step, that is the conversion of cholesterol to pregnenolone in cortisol biosynthesis, which is catalyzed by cytochrome P450 side chain cleavage enzyme, abbreviately known as CYP450-SCC. The cortisol gets metabolized via 11-beta hydroxy steroid dehydrogenase system, 11-beta HSD, possessing two isoforms of the enzyme 11-beta HSD1 and 11-beta HSD2. The former 11-beta HSD1 make use of NADPH and metamorphose it to biologically inert cortisone, followed by final transformation to active cortisol. While 11-beta HSG2 exploit NAD+ to change cortisol. By and large, 11-beta HSD1 dole out to augment the limited concentration of cortisol, and 11-beta HSG2 decreases the restricted concentration of cortisol. Furthermore, cortisol is also metabolized to 5 alpha tetrahydrocortisol and 5 beta tetrahydrocortisol via the catalytic activity of 5 alpha reductase and 5 beta reductase. These two enzymes have been adjudged as the rate limiting factors. 5 beta reductase is also act as the rate limiting factor in the conversion of cortisone to tetrahydrocortisone. It has also been suggested that modification of 11 beta HSD1 play an important role in the pathogenesis of hypertension, insulin resistance and obesity, while 11 beta HSD2 alteration has been implicated in essential hypertension and is acknowledged to escort the syndrome of apparent mineralocorticoid excess. The step-by-step biosynthesis of testosterone can be well understood by the following sequential reactions. The oxidative cleavage of cholesterol side chain by CYP11A, which is a mitochondrial cytochrome P450 oxidase, that trounces the 6 carbon units to bestow the formation of pregnenolone in the endoplasmic reticulum. 2 carbon atoms get obliterated by CYP17A enzyme to yield an assortment of C19 steroids. 3 hydroxyl groups is oxidized by 3 beta HSD to generate androstenedione. Remember, the rate limiting step were 17 beta hydroxysteroid dehydrogenase reduces the androstenedione C17 keto group to yield the testosterone molecule. Especially in men, the testis or the Leydig cells produces high amount of testosterone, more than 95%. Formation takes place in Leydig cells of the testis. On the other hand, in women, a lesser quantity is also synthesized in placenta as well as in thecal cells of the ovaries. The Sertoli cells, which is a part of male generative glands, also requires testosterone for spermatogenesis. So, similar to that of other hormones, in blood testosterone is afforded to target tissues where a large amount of is elated to sex hormone binding globulins, the SHBG, which appears as a specific plasma protein. If we look at the stories of these steroids and its exploitation, we will find some interesting story. In 1930s, the anabolic steroids were first isolated, then identified and further synthesized, and nowadays used in medicine for therapeutic purpose to kindle appetite, bone growth, male puberty, and to take care of chronic wasting conditions like AIDS and cancer. In 1954, the use of anabolic steroids began in sports and games. The 1958 Ziegler, a researcher, hit on methaneandrosterone, which is also popularly known as Dynabol, prepared in the United States by Ciba firm. In 1972 Olympics, there were quite few uses of these steroids, and the range of steroid use was enormous, from 10 to 100 milligram per day. We all remember in 1988 the historical race of Olympic victory by Ben Johnson in 100-meter race. It becomes a popular story as he was found culprit for using the anabolic steroids and failed the drug test. It results in the detachment of the medal he won, and later Carl Lewis declared as the winner. So students, in this lecture we have learned about the biosynthesis of steroids, how the steroids have been regulated and controlled, what are the implications of the cholesterol biosynthesis, the step-by-step reactions carried out in cholesterol biosynthesis, as well as the story of cortisol and testosterone biosynthesis. We will continue with the biosynthesis of lipids, other parts, in the forthcoming lectures. Thank you.